Welcome back to YANE podcast about biohacking and biotech. And today we're going to talk about how do you become a biohacker. So it's basically going to be a guide for people who want to start biohacking. Maybe they don't know how to do that, or how do you start your own research, who is a biohacker, etc., etc. And I want to talk about, well, who is a biohacker? What is biohacking? And I put out some of the dictionaries, and I thought it would be interesting to see, uh, what other people think about what is biohacking. And according to Cambridge Dictionary, biohacking is a scientific experiments with biological material, especially genes, done by people who are not official expert or scientists, either as a hobby or in order to make money or commit crime. I thought this is an interesting one because never heard of anyone who actually committed crime by doing biohacking, but whatever. But there's one that I like more, and it's by Merriam-Webster Dictionary, and the definition is biohacking is biological experimentation as by gene editing or the use of drugs or implants done to improve the qualities of or capabilities of living organisms, especially by individuals and groups working outside of traditional medical or scientific research environment. And I would say that this last definition is more close to what I think biohacking is. And in my opinion, we need to change something in that definition, and is that it says that it's people outside of academia. And in my opinion, biohackers can be someone who's literally had no idea what biology is, starting from um, this perspective that a person never had experience in biological fields and wants to start biohacking. And it can go as far as people who actually have PhDs and done some serious research that are also biohackers. So the term hacker is used in the original sense of finding new and clever ways to do things. And usually when people say biohackers, there's different kinds of biohacking. And some people uh, say that they're biohackers and they're more like grinders who use implants, and it's more of like body hacks culture. And there's the last type of biohacking that I don't like personally. It's very fraudulent in my opinion. It's stuff like bulletproof biohacking, and it's like how to hack your sleep, how to hack your body using, I don't know, bioactive supplements, I don't know, sleeping like with some weird stuff, using nootropics, using microdosing, and different stuff. Most of all because usually it doesn't have any scientific ground. It's just someone, some people do their classes, do their courses, they earn a lot of money by doing that. And I would say that 99% of that is fraudulent, and I highly do not recommend people doing that. Um, in my opinion, it is more dangerous than what we're doing. And I would consider myself a biohacker, a DIY biologist, whatever. I do use term DIY bio a lot, but I like to use biohacker to kind of, in my opinion, try to cleanse the term biohacker. Because a lot of people have negative opinions towards it just because it was misinterpreted, or they would hear about biohackers who are hacking their bodies with intermittent fasting and nootropics. So I want to say that biohacking is science. It is based on scientific approach and actual research. Let's start by that. And so now that we covered what is biohacking in this podcast, I want to tell my story of how I became a biohacker. So I was kind of always interested in biology since I was a kid. I just really liked it. I didn't really like mathematics or stuff like that, and biology really correlated with my ideals, and just I really liked it. Since I was a kid, just like playing in the garden and looking at all the different bugs and flowers and stuff like that. But then when I was graduating high school, my exams were coming, and I was thinking about, well, who should I become? And I was like, okay, well, I really like biology, but I wouldn't be a zoologist or I don't know, ecologist. I wanted to do something more like a hands-on science, something very practical, very useful. So that's why I chose biotech. And I applied to different universities in Ukraine, and the good ones I didn't get into because I didn't have good enough grades for my exams. So I got into Dnipro National University. I was born and raised in Dnipro, so I was like, well, okay, this is the university from my city. It's okay, I'll go. And I was so inspired, by all the scientists, and science was always like magic to me. My superheroes when I was a kid were scientists. And so I was like, oh, I'm gonna become a scientist. I'm gonna change the world, and yada yada yada, and blah blah blah. So I get into university, and well, I was very naive. I mean, as most young people are. I still am naive, right? I'm still young, and I thought that, okay, I'm gonna get into university. I'm gonna start doing research. I'm gonna learn so much stuff. I'm gonna help change the world. And then you kind of hit a brick wall of education, of academia. And I wouldn't say that specifically my university was bad. There's a lot of problems in science, like fundamental problems with publishing, with research, and just academical structure. And if you want to learn more about that, Josena wrote a great article about why scientists are destroying science. And I recommend reading it. I'll put it in description. I'll put it somewhere here in the video. And so I got into university. I was so, I didn't have any knowledge, like just basic high school knowledge. I was like, okay, I'm gonna get into university. I'm gonna learn so much stuff. Then you meet professors who aren't really interested, and you're like, yeah, you're like, I'll wash the test tubes. I just wanna, see the magic happening, see the science happening. Just let me breathe onto it, which is kind of bad metaphor for microbiology, microbiology research. But yeah, well, basically, I hit the wall, and after my first semester, I was really disappointed. I was really depressed because I thought that I had all this potential to do something to change the world, but I couldn't. And then my brother, actually, who's not in biology at all, he told me about Biohacker. So he sent me a few links to different videos, different articles about people who do science in their garages, in their bedrooms, and community biospaces like DIY biolabs, where you can come and there's equipment, everything needed. You can learn, you can do your own projects. So it's like a makerspace, but biotech oriented. And I thought that it was very interesting. At first, I kind of thought it was crazy because I couldn't believe that, well, a lot of people believe the stigma that research can be done only in the super expensive labs by super professionals who've been, I don't know, just, yeah, magic again. So, and my brother, he actually gifted me like a little kit for Christmas, and it was mostly like a children's kit, like for kids to learn how to do biology, how to do genetic engineering. So the kid had like the little transformation station. It was like a little genetic engineer lab. You have like a heating station, cold station, a little incubator. So you could do a bacterial transformation and see them change color and express fluorescent genes and all the fun stuff. And when I first did it, I was, well, absolutely shocked. First of all, because we didn't do that in university. I was like, how is that possible that we can't do this in university? Because it's just, you don't, when you ask professor, they're just like, you don't get it. You just can't. But then you do it at home, and like basically started doing it in my brother's bedroom because he moved out. So that's how I started equipping my own lab. And then kind of step by step, then I found out about the Odin company, and I knew about CRISPR by that time, and it was still very fresh. It was 2017 when I started, 2017, 2018. So CRISPR is still kind of a hot topic. And I really wanted to try it. And the Odin, they started selling these CRISPR kits where you order a kit for like $160, $170. You order it, it arrives, and you have everything you need to do to conduct an experiment and transform bacteria with CRISPR. But the problem for me was that I was a broke student in Ukraine, and $170 was actually a pretty big sum. So, but they had like a, I still do, I still think they do have this, it's not an ad, it's basically like, if you post on Twitter and other social media just with the hashtag biohacking, like you're not even promoting the company, where you try to do different experiments, or you maybe just post your thoughts and you just kind of share with people the idea of biohacking, you earn $2 credit for every post. And I was like, okay, my mission begins. So I started doing all the silly little experiments that I could with all the budget, all the means I had. And I did eventually get a free CRISPR kit. And that's how the story of me getting introduced to the Odin and Joe Zahner and the amazing team went. And then I got invited to Biohack the Planet, yada, yada, yada. By doing that, I was like, okay, this is amazing. I can do this at home. I'm learning so much more than I did at university. And I was studying biotech. Like literally biotechnology and bioengineering is my specialty. So I was like, okay, if I can learn this, other people should. Other people should have the ability to learn stuff like that. So I started inviting my friends from university to, every Wednesday we would gather in my little lab, my brother's room, and we would do little experiments. I would be like, okay, let me teach you how to do CRISPR, and we would just follow kids. And then I thought, okay, I knew about DIY labs. I was like, there has to be one in Ukraine. Spoiler, there wasn't. So I was a little bit disappointed, but then I was like, this is my responsibility to start a DIY lab to educate people, to have people learn and experiment and learn something new, bring knowledge to other people. And for me, it was the ability to give back to Ukraine. And also, our science kind of sucks in Ukraine. It's really underfunded, like majorly underfunded. So for me, it was a way to give a boost to Ukrainian science. So that's how it all started for me. You maybe have different motivation, and that's okay. But I think that's obviously okay. It's great if you are interested in biohacking. So okay, now you decided that you want to be a biohacker. You want to start doing DIY biohacking, whatever you call it. Citizen science, some people call it. And there's numerous names for it. But I call it biohacking because I like it. I started as a biohacker and I still am a biohacker. So some people stumble across, well, I don't have a degree. Daria, you have a degree. You studied, even though it was biotech in Dnipro National University or whatever, still have a background. It is not necessary, in my opinion, at all. Because, first of all, with online education, you can do so much. Okay, you don't have any idea. Maybe you have some basic knowledge about biology from high school, from school. But you have people like David Ishii, who he didn't even graduate high school, and he's one of the most brilliant scientists I know, and he just taught himself that. And you should start small if you want to learn. Just watch videos about how translation happens in DNA, how DNA replication happens, how protein synthesis works. Transcription and stuff like that. I will list a playlist of little videos. They're 2 minutes, 3 minutes long, and it's basically an animation of how molecules work in our cells. And it's very educating, honestly, because when I was studying it in university, I was like, this does not make sense, just because I couldn't visualize it. And for me, it's very important to visualize to understand. I cannot understand just by reading. I need to see how it's done. And I was having my final and I couldn't understand the DNA replication step by step. I just watched a video, 3 minute video, and I came to understanding of how it works. So I have a little playlist of YouTube videos that will help you understand the molecular processes happening in the cell, which is important because biohacking is not necessarily always about genetic engineering, but it's still it's good to know how DNA works. I feel like it's good to know for everyone because if you don't know, then you have people screaming GMO bad, and you go to at a best store in Dnipro and you have toilet paper that has non-GMO on it, and people would be like, "Oh yes, thank God it doesn't have GMO." Um, so, okay, well, you watch those videos and you're like, I want to know more before I start doing something. Because you can always start, but it's better to have like a little basis, just a little bit, so it's easier for you to learn. There's stuff like lectures on YouTube, there's open courses from MIT, and I'm listing the stuff that I used to learn. Um, MIT open courses are pretty good. There's Introduction to Biology. There's a lot about molecular biology that's very useful. There's also a resource like Coursera. They have free courses, they have paid courses, but for people who are Ukrainian students, Coursera is now completely free. So like all the courses are free, and I am currently using Coursera with my not expired student email. I recommend that. So you just learn a little bit, you have a little base. Well, now you want to learn hands-on science, right? You're like, okay, I understand how it works in a cell, but I want to do something. I want to make cell change. So then I would say kits are a great tool because again, I'm just listing what I have done to learn. And there's kits at the Odin, like CRISPR kit, there's human cell kit, there's so many kits. I started with CRISPR kit and bacterial transformation, which is like a jellyfish kit. And this is a great introduction to, like, I think, one of the most used methods in molecular biology, which is transformation, which is how you make a cell, bacterial cell, take up DNA, how you change the DNA in a cell, which is kind of like a basis. And I think it's really good. It comes with everything you need. You don't need to set up a lab yet. You don't need a fancy space or anything. You order it, you have everything, including like gloves, and you just follow protocol step by step. And it's very fun to do. Takes like, I don't know, a few days to finish the experiment, and it's a very rewarding feeling when your experiment works and you see your first genetically modified colonies. It's very satisfying. And so you kind of learn something on Coursera, on like YouTube, stuff like that. Now you're like, I want to start my own experiment. I want to start my own project. Because kits are great. They're great for educational purposes. But well, you can't change the world by completing a kit, right? Completing a course. You need to do something more. And I can't advise you on what topic to choose, what do you want to do. I would say start smaller, like just start small, start simple. And maybe you're more focused on environmental issues. Maybe you want a bacteria that synthesizes some fun protein. Like there's an open insulin project, and they try to make affordable insulin by transforming bacteria and making them synthesize insulin. So an easy project kind of would be like find a protein, find a DNA sequence you can put into bacteria and make it synthesize as a fun protein. Uh, there's, I don't know, you can make yeast synthesize vitamins and stuff like that. Try that. And for example, okay, you need to set up a lab now if you want to do that. And setting a lab can be confusing, and because if you Google DLI.bio, you'll find a lot of garage space and stuff like that, but you also will find a lot of fancy labs. and you would feel like, okay, now I need to gather all of that. But try to focus more on your project and what is very essential for your project. And scientific equipment can be very pricey. And this is why there's a lot of, first of all, there's a lot of open source. There's a lot of open source equipment you can build. There's a lot of stuff you don't actually need to buy. And you can just Google that. I will list all the resources for that. There's Hacketeria, there's Open Wetware, there's a lot of there's people like Sebastian Kosioba who build a lot of DIY equipment. Joe Zayner, David Ishii, we all did that. We all had to improvise. So I'll say the basic for your lab would be an incubator, which is not actually a super necessity. You can just leave your bacteria at room temperature. It will just take longer to grow. You can just put it in a warm place, put it on a radiator, and etc. You will definitely need a pipette. This is the basis. This is pipette is your weapon as a biotechnologist. You have to have a pipette. Different regions. There's a lot of places to get regions. eBay, Amazon, Deodin. There's lots of different places. And you need media for your bacteria to grow. You obviously need a lot of plastic, like petri dishes and test tubes and stuff like that. And then coming more into like molecular bio, you can, PCR machine is also very, very useful little thingy. Not little, but it can be little. There's a project called Gaudy Labs, and they have pocket PCR. So now we all know what PCR is because of COVID. Pocket PCR is this big. It literally fits in your pocket, and it's a little machine that amplifies DNA. So, for example, you had 1 copy of DNA, then after 30 cycles, you have 3 billion copies of DNA. So it's like a little printer for DNA, which is very useful for different stuff in molecular biology. And there's a lot of guides. There's a lot on the internet, let's say. There's a lot of resources, forums where you can learn. You can also look at other biohackers, see what they're working on. You will see that different people work on different stuff just because different stuff interests them. Some are working on engineering plans. Some are working on making cool pigments. Some are working on just popularization of science, right? Science communication, which is not also bad. And so choose your topic. Choose what do you want to do. Start small. You may want to solve a global problem, cure cancer, change global warming, stuff like that. But don't get me wrong, it's a great idea, it's important. But in order to do that, you need to start small. So scale down your experiment. Start by doing something small. And next, how do you plan your experiment? Okay, you have, for example, chosen a topic. You have a problem that you want to solve and you think that you can do it. Well, first of all, look into what scientists have done, right? Look into the scientific papers. And there is a problem that there is a paywall behind most of the scientific papers. This is why you use Sci-Hub, our best friend. Sci-Hub is basically, it's a Telegram bot. It's a website where you can just Google DOI, and DOI is like an identification number. It's unique for every paper. This is how you find a paper. And just type it in there and it will give you a paper, mostly. Not all of them are there, but the most popular ones, the most recent ones are all there. So read papers about what other scientists have done. What have they accomplished? What do you think they missed? Because sometimes scientists miss, or there's just such a broad, area of study that maybe science is just, they look into like 10% of it. They haven't looked at it in different organisms and just haven't used different methods and stuff like that. So read, and you have to read a lot of scientific papers. And scientific papers are not an easy literature. It's not something you read before you go to bed. It is a little complicated, but start small. You know, start find a scientific paper like 1962 Francis Crick James Watson about how they discovered the structure of DNA. Read that paper. You can start reading papers by reading some basic papers, not basic, I mean they're fundamental, I meant, about major scientific research that led to stuff like genetic engineering. And it will help you understand scientific language and just how scientists interpret their results, because scientists do not use an easy language to describe their work. And after you read a few fundamental papers, start small and start digging papers about topic that is either very close, or maybe it will be very same. And usually there is a lot of research about, even though you may think your topic no one have thought about it, there is probably a paper about it. And after you have read your scientific papers, you can see what scientists have done. What do you want to do? Maybe you want to try something new approach, maybe change something a little bit in what they have done. Write a plan. What do you need to do step by step? Then write a list of equipment, of regions and stuff like that you need, and write a timeline, rough one, because timeline, because science doesn't always work like we want it to work. Sometimes it's like magic, like doing PCR. I swear to God, I think it's magic, because you can do everything correctly. You can do everything like in a protocol, and it's still not gonna work. So you will have to improvise a lot. Sometimes you'll want to hit your head on the wall, but it's just how science is. It's not for mentally well people. It's for masochists, in my opinion. And do that. And you can always also ask people. You can literally email people, for example, from the paper you're using as a base for your experiment. You can try to actually contact them if they're still alive, the people who wrote the paper. You can find their emails and just try to be really nice. And not all of them, but some do respond. And Google, and do your research. And then your first project will help you set up your lab, kind of build a scaffold of what can be built on top of it. And what next? You can do your project, but why, right? And the sky is the limit, right? What's next? The sky is the limit. You can start your own biotech company. You can make your own DIY biospace and gather people. You can, I don't know, make a cure for cancer, theoretically. Like, there is so many things you can do. You can do art with biohacking. Just honestly, it's limited only by your creativity. And it's science is art, and art is science. So we may think there's a lot of limits in science, but I honestly think that it's so many things were unthinkable, right, by scientists. And even atom, right? People thought that atom is a fundamental particle, right? It was named, like the Greek word or the Latin word is unbreakable, undividable. But then we learned that no, atoms actually can divide and stuff like that. So even though you will read a lot of scientific papers, and the academia is it has produced a lot of useful things, but don't let people limit you in what you want to accomplish and what you want to do. Don't let old white guys kill your dreams and kill your passion. Just keep that light in you, and it will lead you to a lot of greatness, in my opinion. And always ask for help. You can ask me for help. You can reach out to other biohackers if their research is relatable to your research. And just do stuff. Honestly, the best advice I can give to you is do stuff. You will mess up a lot, but you will learn so much. It's a great journey for a human to start. And this is this is I think everything I wanted to say. And if you want to support YANELab, if you want to support our team, please donate to our Patreon. If you want to support Ukraine during this hard time, I also put a link down on how you can support Ukraine. Even doesn't have to be a great sum. It can be basically your cup of coffee in the morning, and it will do a lot of good. And also, not sponsored, but it could be Joe Zahner. You can use code YANELab for 20% off at the Odin and get your supplies there and learn. And see you next time. Good luck with becoming a biohacker.